Okay, well, it's 11 o'clock, at least in my time zone. So good morning or good afternoon, depending on where you're joining from. My name is Eva Birnbaum. I'm the program manager for the Isotope Program at Los Alamos National Laboratory, and I would like to welcome everybody to the Actinium-225 User Meeting. Just a reminder, this is being recorded, and we will have obviously a great panel of speakers here today. I will kick off with a short introduction, and we will have a lot of time for questions at the end. So I will encourage people to put questions into the chat so we can answer them later if you have things that come up during the presentations. And otherwise, certainly we look forward to discussion at the end. So with that, I will go ahead and share my screen and kick this off. All right, well, great. Well, welcome again to the DOE Isotope Program User Group Meeting. I am giving this from my perspective at Los Alamos National Laboratory, but I also wanted to acknowledge Dan Strassner as the overall TriLab project lead. So again, thank you for joining today. I'll go through some brief history on supply and demand and our accelerator-based production route for Actinium-225, the status of the DOE Isotope Program Drug Master File development, improvements that we're pursuing within the program, and then we'll move to our roundtable and our different panel presentations, followed by a moderated Q&A session. So certainly I wanted to start just by recognizing the significant amount of recent clinical experience with both Actinium-225 and bismuth-213 labeled compounds. And you can see here a great summary from a recent manuscript from Morgenstern and et al., just describing really the large number of studies that have been recently conducted using actinium and bismuth to treat a variety of different diseases, including cancer as well as others. And really Actinium-225 DOTA compounds with the PMSA617 ligand have really demonstrated the efficacy of this route and is paving the way for a lot of new applications that I expect to hear more about later today during these presentations. So from a supply and demand perspective, the current worldwide supply of Actinium-225 is on the order of 1,200 to 1,700 millicuries per year. And the patient doses that have been informed through these recent studies are on the order of 160 to 640 microcuries per patient for Actinium-225, and would require generator loadings on the order of 100 to 150 millicuries of bismuth to enable the bismuth-213 routes. And so these projections then lead to an assessment that we really need to increase the supply of Actinium-225 to enable broad scale application of both of these isotopes. From the DOE Isotope Program, the Oak Ridge supply has been a major contributor to enabling these initial studies. And this has all come from the decay of existing thorium-229 stock. So over 10 curies of Actinium-225 have been shipped in over 2,000 different packages. We can supply approximately 1 curie of Actinium-225 each year via this route. And we have conducted 13 four-week campaigns during the last year with weekly customer shipments. And so this has certainly enabled significant discovery activities to continue, but this supply is limited and insufficient to meet the growing demand for Actinium-225 and bismuth-213. So therefore, we've been pursuing for a number of years now an accelerator-based production route to increase Actinium-225 supply. And you can see in the upper left-hand corner the original cross-section measurements that were collected from within the program to inform this. And these now are have been translated into IAEA recommended cross sections for the accelerator-based production route. across the energy that we have available for this. And from Los Alamos and Brookhaven as the two DOE Isotope Program Accelerator sites, we anticipate that we can supply between 1 and 2 curies per target with a 10-day irradiation to increase the availability of Actinium-225. And facility investments at both the isotope production facility at Lannel and the Brookhaven linear isotope producer have increased our projected production capabilities. And this became the basis of the DOE Isotope Program Tri-Lab effort. So utilizing the expertise and capabilities from these 3 sites to increase the supply of Actinium-225. So at Los Alamos, we have the isotope production facility where we can take a 100 MeV proton beam at proton currents up to 275 microamps for routine production. The Brookhaven linear accelerator facility as well can have 165 microamps on target and a broader range of energy between 66 and 202 MeV. And then coupling these production sites with the processing experience and capability at the Oak Ridge National Laboratory to continue to supply Actinium-225 to the broad user community. Accelerator produced Actinium-225, as most of you are probably aware, does come with co-production of Actinium-227. The ratio is dependent on the instant energy of the proton bombardment, but we understand what is produced and can communicate those levels to the end users. So the Actinium-225 co-produced product does create some challenges. This long-lived isotope can lead to facility licensing concerns, as well as end-use concerns with patient waste disposition. But these sorts of challenges are not unique and have been addressed for other isotope programs. Conclusions from initial studies of accelerator-produced Actinium-225 are that it does perform similarly to the actinium that is derived from thorium-229. And the direct labeling efficiencies are compatible. The generator performance is similar. And the impact of the Actinium-227 on dosimetry has been demonstrated to be small. So we continue to work to address the challenges, though, that are associated with the Actinium-227 product, as listed here. Updates on the status of the DOE Isotope Program Drug Master Files. A Type 2 Drug Master File was submitted to the FDA in December of 2019 for the accelerator-produced Actinium-225, and that has been acknowledged to have been received. A similar DMF was submitted in December of 2020 for the thorium-229 derived Actinium-225 product. The program continues to interact with the FDA in reference to both products to ensure that we continue to move forward with these. And we're really committed to making these products available to our customers and the user community, and are happy to address any further questions on the status of the DMF. We are also continuing to work to increase both the frequency and the size of accelerator-produced Actinium-225 batches. So we are addressing the technical and logistical challenges while ensuring that our product is consistent and reliable. Additional facility investments are in progress to enable these more frequent and larger batch sizes, and we're building in processing capability redundancy to enhance reliability of this product. We are also continuing to pursue alternative routes of production. We won't go into detail on these today, but I list them here. Electron linac production, low energy cyclotron production, or additional thorium-229 production via reactor to continue to give depth of supply for these important isotopes. I also wanted to highlight a new isotope under development. Cerium-134 is a potential F element PET imager that is chemically similar to either actinium or thorium, depending on how the oxidation state is controlled. And so this is a very nice new isotope that can be used to help facilitate a discovery and development of Actinium-225 as a more complementary PET imager. And Rebecca Abergel is going to talk about this isotope additionally later this morning. So we have evaluation batches that are currently in progress on this, and we hope to make this isotope more widely available in the near future. So in summary, the TriLab effort is routinely producing Actinium-225, and product is available for end users. We've distributed over 440 millicuries of the Accelerator boost Actinium-225 product, and we're working with companies and hospitals to continue to support phase 1 trials and have seen increased adoption of the Accelerator-based product. We will continue to work to facilitate understanding of the Actinium-227 content and make sure that regulatory challenges are addressed. And we're continuing to scale up availability of this very important isotope. And so with that, I will conclude this introduction and we will move to the next presentations. And so I look forward to hearing first from Rebecca Abergel from the University of California, Berkeley. Let me quit sharing my screen here. And so she will be talking on developing new tools to facilitate the end use of Actinium-225. So Rebecca, please.
Okay, so hopefully you can see this and it's the right display. All right. Good morning, everyone. Thanks, Eva. I am honored to be here and to talk to you all a little bit about what my group is doing, not necessarily only with Actinium-225, but and what we're trying to do to facilitate the use of Actinium-225 and more specifically Accelerator-produced Actinium-225. And that include developing chemical tools, new isotopes, but also new counting methodologies or approaches. So if you dialed in this meeting here, you probably are very familiar with this decay chain already. So I'm not going to show anything new to anyone. Just to highlight again that Actinium-225 has several alpha particles along the decay chain here before getting to stable bismuth. It is about a 10-day half-life, and there are a lot of specificity about the chemistry of actinium that makes the use of this isotope especially difficult for biomedical applications. And that includes some of those listed here, which would be that the kinetics of complexation with some various chelators are very slow. Most chelators that we have on hand will form complexes that have pretty low thermodynamic stability. Also, actinium, unfortunately, behaves very differently from other +3 actinides, the trivalent actinides. And we don't know so much about its chemistry because the only 2 isotopes really available are Actinium-225 and 227. Their half-lives are very short. We don't have so much to do any real bulk chemistry. And so we do have very little chemical information, which makes it difficult to develop new chelators. And on top of this, Actinium-225 is spectroscopically silent. Mostly, there are no real useful gammas coming out so that we could image actinium easily. So we're trying to address some questions that are related to those hurdles that come with the use of Actinium-225. And one additional hurdle is that the Accelerator produced Actinium-225 may come with a little bit of Actinium-227, as Eva pointed out, which is a much longer half-life and also goes through a very different decay chain in which you'll find also a lot of alpha particles emitted before it decays to stable lead. So among the questions that we're trying to ask and potentially answer are there easier ways to quantify the amount of Actinium-227 in Accelerator-produced Actinium-225? So when the user would receive a batch of Accelerator-produced actinium, could there be an easy method that you can implement in every lab to really quantify the amount of Actinium-227 and just to make sure that we can produce viable formulations that can then be injected into animals or humans? A second point would be, how can we develop methodologies to perform precise in vivo imaging? So, what surrogate isotopes can we use to use standard PET or SPECT methodologies so that we can actually follow the actinium bioconjugates in vivo and really look with image the evolution of the disease or how well those antibodies are working. And the last point I wanted to bring up is, can we develop new bioconjugation methods that will lead to highly stable complexes that will ensure that the actinium is actually bound to the targeting moiety, a peptide, or an antibody, and that can be achieved with fast binding kinetics so that you don't have to worry about increasing the temperature during labeling and potentially degrading the biological vectors. So I'm going to go very quickly through what we were trying to do to address those 3 points, and hopefully that will give some ideas. The first one is led by a graduate student in my group, Cathie Shields, and she's really spent a lot of time looking at this decay chain. And most of you, if you have thought about Actinium-227 as a contaminant in Accelerator produced Actinium-225, have probably looked at the main decay chain, which would go through thorium-227 and radium-223, which are both alpha emitters, but have very long half-lives. And so on the order of 18 and 11 days. And that means to get to circular equilibrium would take months after separation and purification of your actinium product that contains Actinium-227. If you look carefully at this decay chain, you'll also find this 1.36% alpha branch that goes to francium-223. And francium is a beta emitter that can actually get to secular equilibrium with Actinium-227 within hours. And so if we were able to quantify francium-223 in our samples, we could actually quantify with high precision the amount of Actinium-227 in the Accelerator product at different time points as along the purification and handling with other chemical species, including bioconjugation. And so you're looking at the decay here, so that's the alpha branch that we're talking about, and potentially we're looking at this gamma lines here around 50 keV. And so if you're looking at a gamma spectrum of francium-223, you'll see that it decays very quickly because the half-life is very short. However, that allows us to really quantify the amount of francium-223 and the amount of Actinium-227 it was eluted from on a simple column. And so this peak here, around 50 keV, does not overlap with any other line coming from Actinium-225. And so assuming that the separation from the thorium target in the production of Actinium-225 is high yield and you get an actinium product with decent purity, then we really have a signature here that allows us to quantify Actinium-227. And so hopefully this will be published shortly, and that will give a methodology for everyone to be able to quantify the contaminant in their batches. The second point I wanted to address is the use of other isotopes as surrogates to help with in vivo imaging. And so here we're looking at everything available out there. We're focusing on Actinium-225. All of those elements here highlighted in green are those elements that have alpha emitting isotopes of interest for targeted alpha therapy. And so you see that actinium here is the first element in the series of the actinides. It's a very large cation, +3 cation, the largest one that we have here if we look at all those metals. And so chemically speaking, it is very unlikely to behave similarly to some of the PET isotopes that we have on hand, including scandium-44, yttrium-86, zirconium-89, or gallium-68. For example, actinium and gallium have a very different chemistry. Gallium is a transition metal. It's much smaller than actinium +3. Scandium and yttrium are rare earth, so they may be a little closer in terms of chemistry, but still much smaller than actinium. Zirconium is a +4 oxidation state. So again, those 4 potential imaging probes are not very likely to behave similarly to actinium. And so a few years ago, we started exploring the potential use of cerium-134 as an isotope that could behave similarly to actinium, and also as an isotope that could behave similarly to thorium, and that thorium-227 is an alpha emitter of interest. And the rationale behind this is that the ionic radius of cerium +3 is very close to that of actinium +3, and the ionic radius of cerium +4 is close to that of thorium +4. And so in a collaboration with Stash Kazimor and Etienne Fermolin at Los Alamos National Laboratory, we've started exploring the use of cerium-134. It's generated through a P6N reaction from a lanthanum target. And just to highlight here is that cerium-134 decays very quickly into lanthanum-134, and lanthanum-134 is the actual PET emitter. But what we believe is that we can use cerium-134 then as a PET surrogate. And so here is a study where we compared 2 metal complexes formed with cerium. The first one is with DTPA, so a standard chelator that will form a cerium-3 complex. And so those are PET images of the cerium-134 complex with DTPA with very fast excretion through the kidneys and the biliary output. And after 24 hours, you see that it's all excreted, so a very stable complex. And then on the bottom line, we're looking at another cerium complex formed with a hydroxypyridinone chelator here, the 3,4,3-Li1,2-HOPO. And the particularity of that chelator is that it oxidizes cerium to cerium +4. And so that would be a good surrogate for thorium-227 since it's in the +4 oxidation state. And what you see here is now excretion through the biliary pathway, through the liver, but also again, fast excretion, 24 hours, it's mostly gone. And more recently, we've also used DOTA as a chelator for cerium-134. And here, those are images from a targeted study where we targeted HER2 positive tumors with trastuzumab attached to DOTA and cerium-134, and you see accumulation in the tumors fairly quickly. And so looking at ex vivo distribution, we show again that the cerium-134 distribution is very close to that of actinium when it's cerium +3, and very close to thorium when it's cerium +4. Now, the last point I wanted to bring to your attention is that radiolabeling biological vectors with actinium can sometimes be very tricky, especially if we're dealing with antibodies, in that we have to very slow kinetics, and so that means we have to heat our samples at pretty high temperatures that may degrade the antibodies. And so we've started trying to develop some new chemical tools that would facilitate the radiolabeling through either click chemistry or some non-covalent bonding interactions. And so in this case, I'm focusing on this hydroxypyridinone chelator that has been used in various antibodies with zirconium by the group of Francesconi. But this methodologies could apply to pretty much any chelator, and that also applies to actinium. And so the idea here is that we're making constructs in advance that include a targeting moiety. In this particular study, we're looking at transferrin, but we can use any antibody. And in this case, we're attaching that to mesopore silica nanoparticles that have pores that can absorb the chelator, and the chelator is shown here as the circle. The pores are then blocked with some cyclodextrin moieties so that the chelator is trapped inside the particles, and then you soak this into your radioisotope solution that forms the metal complex inside the silicon nanoparticles. So you can really load them up with radioisotopes without having to do any kind of high temperature labeling or subsequent antibody attachment. And so this kind of methodology is hopefully opening some path to the use of different chelators and different isotopes with preconditioning with different targets. Another way to think about this is using other proteins. And so in this case, we're using non-covalent interaction between a protein called siderocaline that's attached to antibodies in advance. And that protein has a calyx that will then bind through electrostatic interactions, a chelator radionuclide complex, and again here we're looking at an actinium hydroxypyridinone complex. And so again, this methodology allows us to prepare in advance our fusion antibody constructs with the chelator, soak that in a radioisotope solution, and leveraging fast kinetics, low temperature, and high stability, having the radioisotope labeled quickly. So with that, hopefully I didn't go over time. I'll just point out that this work is done by graduate student and postdocs in my lab. Cathie Shields is leading the francium effort, Tyler the cerium effort, Dalia does most of the animal work, and Roger led the nanoparticle efforts, and Matt and Josh participated in all animal experiments. The cerium 134 work was done in collaboration with Lano, Stosh Kazimor, and Etienne Fermelin, also including Veronica Moko, and also together with John Engel at University of Wisconsin-Madison. Our protein work is done with the Fred Hutchinson Cancer Research Center, and we're leveraging very nice facilities at the Lawrence Berkeley Lab for imaging. And I'm happy to answer. I should mention that parts of this work were funded by the Isotope Production Program, the DOE Office of Science, and we get most of our isotopes through NIDC. And I'll be happy to answer questions later on.
Thank you, Rebecca. And yeah, I'll just remind everybody, you can put questions into the Q&A to be answered at the end, or we'll have a more open forum. So we'll go ahead and move to our next talk. Ekaterina Dadachova from the University of Saskatchewan is going to speak now on Actinium-225 and Bismuth-213 for cancer, infections, and beyond.
Okay, thank you very much, Eva, for a nice introduction. And I would like to thank the organizers for inviting me to speak and be part of that panel. It's a great honor and great pleasure to talk about our work with actinium and bismuth. So before I go into highlighting some of our projects, okay, I would like to draw your attention to the very recent review which just came out this month. It emanates from the Lawrence Livermore lab, published in Coordination Chemistry Reviews, and basically they analyzed the old data which we all fall onto from the Shannon paper and others, and then they analyzed the data from the 70s from EXAFS, and also performed their own investigation and research, and they demonstrated that actinium cation is a bit smaller than we all thought, which is a very interesting, I think, piece of information, and might explain, for example, while when we label with actinium, it's not supposed to fit well in daughter cavity, but for some reason it does. Our radiochemist in the lab, Dr. Kevin Allen, who was talking at that workshop last year, can achieve 99% labeling of daughter with actinium conjugated to the antibodies at 37 degrees. That always surprised us. And on the other hand, when back then I was a trainee in Martin's Brechbill's lab and had that privilege of being part of his group, we were designing different larger macrocyclics, and sometimes actinium was just slipping out of the grip of those macrocyclics. So I think, again, this review informs the field about that interesting ever-evolving property of actinium, and will help to design better chelating agents and better therapeutics based on actinium. So today,
I apologize for interrupting. Can you put it into full screen? We're seeing the slides on the slide.
I will try, but last time my computer didn't allow me to go into like rewind forward in my slides. I will try. Just a second. Okay. Can you tell me when I move to next slide if you can see it? Okay.
Yeah, I can see it. Thank you.
So I will talk about 3 projects in our lab where we use actinium as is. then we use bismuth, and then we use actinium and bismuth side by side. So the first project is done in collaboration with Actinium Pharmaceuticals, an industrial partner which has been working in the clinical space for a long time, conducting clinical trials with actinium-labeled antibodies. So here we investigated the interaction of a CD33 targeting lintuzumab antibody labeled with actinium and BCL inhibitor venetoclax, which has been approved a couple of years ago for treatment of acute myeloid leukemia. Can you see next slide, Eva? Can you see the graph?
Yes. Yes.
Okay, great. So it means it's advancing. Thank you so much. This is the combination study in mice bearing xenografts with one of those cell lines, acute myeloid leukemia cell lines, OCI AML3. And as you can see here, the lintuzumab, if unlabeled, doesn't do much to this aggressive tumor. And venetoclax by itself also just a little bit slows this down, but not much. Actinium-labeled lintuzumab, on the other hand, actually suppresses very significantly the growth of those xenografts, and the combination of actinium labeled lintuzumab with venetoclax leads to complete abrogation of tumor growth. And this is also nicely reflected in the overall survival of the mice, which was 80%. So what the mechanism of this action, actinium lintuzumab basically lowers the levels of many anti-apoptotic proteins in the acute myeloid leukemia, such as MCL1, BCL2, and BCL-XL. And this plot shows you those levels going down as the activity. This is kilobecquerels of actinium-labeled antibody, as activity of actinium-labeled lintuzumab is increasing. Also, what's going on, we saw that there is much more DNA double breaks induced by lintuzumab, actinium-labeled lintuzumab, than in untreated cells, and specifically the combination induced many more double DNA strand breaks than in any other treatment. And this, I think, leads to really nice, impressive results. This study was championed by Dr. Ravindra Garg, a research associate in the lab, recently published, and Actinium Pharmaceuticals is currently conducting a clinical trial of that combination therapy. So that's how actinium by itself would be very effective in certain indications, such as acute myeloid leukemia. So now it's a different topic. I will talk about bismuth-labeled antibodies for something which we have been doing in the lab for a long time. This is radioimmunotherapy of fungal infections. We know that there are certain fungal infections in immunocompromised people which could not be treated with antifungals because the immune system of such patients is not working. And recent very sobering examples, we see that in patients with COVID, for example, in India, they were infected with those black fungi, mucor and others, and those infections could be really devastating. So years ago, in 2012, we published a position paper where we stated that there are certain antigens expressed by every pathogenic fungus which infects humans. So if you have radioimmunotherapy with the antibody directed towards one of those universal antigens, you can treat a patient with any opportunistic fungal infection with radioimmunotherapy. And here is the list of those universal targets. One is heat shock protein 60, then it's beta-1,3 glucan, ceramide, and melanin. Melanin is that black pigment which gives many of those black fungi that terrifying black color. So these are all surface antigens which could be conveniently targeted with radioimmunotherapy. And back then, in the same paper, we performed some in vitro killing of those fungi, two very widespread pathogenic fungi, Candida albicans and Cryptococcus neoformans. The descending lines is the killing of the cells with bismuth-labeled antibody to one of these antigens, namely beta-1,3 beta-glucans. But for many years, we didn't have a chance to perform in vivo experiments. So finally, when I was already working at the University of Saskatchewan, we got permission to work with B. dermatitis. This is a fungal pathogen which actually can infect even a healthy individual. It also affects dogs a lot because they dig in the soil and they inhale the spores. It's level 3 pathogen. So here we infected mice with, blastomyces dermatitis, and we then treated them with bismuth-labeled antibody to beta-glucan. And as you can see, the cold unlabeled antibody didn't do much, and this is logarithmic scale, by the way, showing the colony-forming units per milliliter of the lung tissue. And the bismuth-labeled antibody to 1,3 beta-glucans decreased the fungal load by 2 logs. And this is how the plate, when you plate the mouse lungs, this is the lungs of the healthy mouse. This is the lungs of the mouse treated with bismuth-labeled specific antibody. This is non-specific labeled control, and this is cold antibody, and see that plate is overgrown with fungus. This is the untreated mouse. Then we went ahead and performed a safety study in dogs, in healthy dogs and beagles. We just gave them a certain activity, which we recalculated from the mouse doses, and observed those dogs for 6 months. For all kinds of parameters, blood chemistry, liver, kidneys, functions, and everything was normal. Then those dogs went to doing what they were always doing. Our veterinary students just examined them to learn how to examine a dog. So basically that was very encouraging. We were planning to perform a clinical trial in dogs with blastomycosis as a large animal model, but of course pandemic changed everything. It's now difficult for us to get the dogs across the border. So I guess we'll probably will be doing some black fungi work relevant to COVID soon. And the last topic which I want to cover in my talk is using side-by-side bismuth and actinium for radioimmunotherapy of HIV. We know that HIV is now manageable disease thanks to antiretroviral therapy, but it's not curable. There is no cure for that. There is no vaccine for that. And also there are certain problems associated with HIV, people who are treated with antiretroviral, which I will quickly cover. So again, back in 2012, we published a paper in PLOS ONE where we used the antibody, fully human antibody 2556, labeled with bismuth. It is directed to GP41 glycoprotein on HIV-1, and that motif of that glycoprotein is conserved among all the clades of HIV. And we showed in mouse models, which were available to us back then, basically SCID mice injected with infected human cells, that we can eradicate those HIV infected cells by targeting the GP41 protein expressed on the surface of those cells. Then Dina Cukrov, the graduate student in the lab at that time, she collected the blood from the patients who were treated in our Montefiore Medical Center in the Bronx. Some of them were ARC naive, and there were or the two different cohorts of standard art treatment, and then she treated that blood with a bismuth labeled antibody, and she showed nice killing of the infected cells in the blood of those patients. But also there is a huge problem within HIV, which not even some colleagues who work in HIV field are aware of, that there is a problem of NeuroHIV and NeuroAIDS. HIV enters the brain during the first week of infection and it doesn't leave, and antiretroviral drugs do not penetrate the blood-brain barrier, and unfortunate patients, many of them are first developing a cognitive deficit and then they are going into cognitive decline. So these are the photos from that New York magazine in 2009. So this slide is courtesy of my colleague at Albert Einstein, Dr. Joan Berman. So the monocytes are the culprits behind bringing HIV very early during infection into the CNS and into the brain. So what we did back then in collaboration with her lab had in vitro fully human model of the blood-brain barrier. And we were letting the infected monocytes go through the blood-brain barrier, and then we were chasing them with that radio-labeled antibody 2556 labeled with bismuth 213, and we showed that it specifically killed those infected monocytes on the CNS side of the blood-brain barrier, while confocal microscopy showed that this blood-brain barrier was not disturbed by radioimmunotherapy. And Alicia McFarren, a medical fellow in the lab, did that work. So this is the last data slide. It's again the work of Ravendra Garg, who was working with Actinium project. I showed we realized that in, say, non-human primates, which would be the next frontier in this research, if we one day get funded for that. And in humans, of course, we need a longer-lived isotope. And here Ravendra very nicely showed that both bismuth and actinium kill very specifically monocytes in vitro. In comparison, for example, with the control fully human antibody, which is used for treatment of respiratory viruses. So we hope that our next step will be using combination treatment of RIT and possibly reactivation drugs in state-of-the-art humanized mice, which we have a wonderful collaborator at University of Saskatchewan, Dr. Carrie Lavender. She is the person behind that model. And then hopefully we will move into the monkey model. So my last slide, I know that my title stated beyond, but I realized 15 minutes it's too little. So we think that the next frontier for actinium and bismuth could be selectively targeting cytotoxic T and B cells in autoimmune disorders such as diabetes, lupus, and multiple sclerosis. Currently, we have a project in our lab going on in this area, championed by graduate student Connor Frank, and hopefully next year I will present you the encouraging results. So the conclusions are that actinium and bismuth are unique. They're great for cancer, infectious disease, and hopefully for autoimmune disorders, and we all need to collaborate with industry and physicians to bring those novel therapies to patients. So I would like to thank my lab. They are all wonderful, talented, and extremely dedicated people, and multiplicity of collaborators. For the sake of time, I'm not naming all the names, but they are here in the funding from some of the US funding agencies and Canadian funding agencies. Thank you very much for your attention.
Thank you very much, Kate. That was a great presentation. Again, we're holding questions to the end. And so we'll move on now to our next panelists. We have a joint presentation from Fusion Pharmaceuticals, Jim O'Leary and Thomas Armour, talking about single ascending dose phase 1 experience with Actinium-225 FPI 1434 in patients with IGF-1R expressing solid tumors. So thank you.
So once again, thanks for inviting us here today. My name is Jim O'Leary. I'm the Chief Medical Officer here at Fusion. And together with our Director of Molecular Imaging, Thomas Ammer, we'd like to give you an update on where we're at with our first radiopharmaceutics to be brought into the clinic. So we are developing FPI-1434, which is an IGF-1R targeting radiopharmaceutic. Of course, that is radioconjugated to actinium. And for background, IGF-1R, as you know, is a target that has been targeted in the past with a number of naked antibodies as well as protein kinase inhibitors. And it's implicated in a number of functions in cellular proliferation, metastatic potential cell survival of cancer cells. IGF-1R is expressed across a wide range of tumors, which are highlighted in the middle of the slide. And this is just based on review of the literature, so it's really meant to be directional. But as you can see, developing an IGF-1 targeting radiopharmaceutic could have broad potential across a wide variety of tumors. On the right-hand side, we show our imaging agent, which is we call FPI-1547, and in place of actinium, we have the radioisotope indium to produce better images. And then on the bottom half, you see the structure of FPI-1434 with actinium. So our first human study has two parts to the phase 1 dose escalation portion. We started with a single dose administration dose escalation and are currently recruiting patients to the multi-dosing portion of the study. And as typical of any phase 1 oncology study, our primary objective is to understand the safety and tolerability profile of both the imaging analog and the therapeutic FPI-1434. All patients are with solid tumor, as long as they are identified as having positive imaging, are eligible to participate in the study. Of course, we're also looking to identify our maximum tolerated dose and recommended phase 2 dose. Patient selection, as I mentioned, is and individualized patient dosimetry is performed based on imaging with the indium analog. We are employing a typical 3+3 dose escalation design. And as I mentioned, recruitment to the single dose escalation portion has completed, and we are now enrolling patients into the multi-dosing portion, as well as a sub-cohort of the study, which evaluates the impact of pre-administration of cold antibody on the characteristics and distribution of the imaging analog. So key eligibility criteria, as I mentioned, tumors must light up positive on indium imaging. They must have a target to background ratio of at least 2 to 1. So any solid tumor is eligible to participate, any patient with any solid tumor is eligible to participate in the study, as long as they meet that target to background ratio 2 to 1. ECOG performance status and adequate end organ function are typical, again, of any phase 1 oncology study. With respect to administration of prior radiopharmaceuticals, we have at least a 6-month interval from prior treatment, and prior radiation to large areas of bone marrow less than 20 gray. If a patient has more than 20 gray, they are not allowed to participate in the study. We have a safety review committee in place that evaluates each cohort before escalating to the next cohort. And that's comprised of all of our study investigators, as well as some independent people, medical oncologists, physicists, and a nuclear medicine physician. So you can see in terms of the patients that were recruited to the study, median age of 61, a few more males than females, which probably goes hand in hand with the fact that we had a number of prostate cancer patients, 6, compared to colorectal and the other single tumor indications. And this was based on the fact that early in our the conduct of the study, one of our largest sites in Canada was a physician who treats GU cancers, specifically prostate cancer. In terms of the most common related adverse events that have been observed thus far across the 3 cohorts, and I forgot to mention, the 3 cohorts that we evaluated ascending doses of FPI-1434 at 10, 20, and 40 kilobecquerels per kilogram. And all in all, at all 3 dose levels, both the imaging agent and the therapeutic have been well tolerated. As anticipated based on our IND enabling studies, myelosuppression was the most common adverse event. And you can see the distribution of patients who experience thrombocytopenia, neutropenia, and lymphocytopenia. In terms of grade 3 events across these 3 cohorts, we had 1 event of grade 3 neutropenia, grade 3 lymphocyte count decreased, and grade 3 white blood cell count decreased. And all 3 of those adverse events were noted in the same patient. Perhaps most noteworthy is that through these first 3 dose levels of escalation, there were no grade 4 adverse events observed. And across all cohorts, we have not seen any dose-limiting toxicities. We've not seen any treatment-related serious adverse events, and dose interruption or modification were not needed for patients treated thus far. For the dose escalation, first part of the dose escalation study, our DLT observation period was 56 days. This was mandated by the FDA. For the multi-dosing portion, we have been able to decrease that dose-limiting toxicity observation period to 6 weeks. So patients are now eligible to receive FPI 1434 at 6-week intervals with repeat doses of administration, up to the maximum accumulated tolerability limits for organs. And at this point, Tom, maybe I'll ask you to go over the next couple of slides.
Sure, thanks, Jim. So in this study, as Jim mentioned before, we were using the indium labeled analog for imaging. We're doing serial planar scintigraphy at 4 time points over the course of a week prior to the therapeutic administration. And that's to generate statistics for organs and tissues. We kind of work up the kinetics for all those different compartments. We're using CT scans to generate patient-specific volumes for kidneys and other organs as needed. And activity is integrated over time, and we're working on radiation dose estimates centrally using the radar method as implemented in the Olinda software package. Really mostly using the planar imaging over time for the dosimetry piece, and then the SPECT CTs, as Jim mentioned, are used for eligibility of the tumor. Next slide. So just have a couple examples to present of what imaging looks like with FPI 1547. Just example of those 4 different planar scintigraphy time points over time. And this example is a patient, 65-year-old with advanced ovarian cancer. And you can see clearance from the blood pool at about 3 days post-injection in this example. And then the axial SPECT CT slices are presented below, showing the qualifying lesion in this case that the patient presented with. Next slide, have another example. And this one is a 69-year-old male with metastatic prostate cancer. And again, we see early clearance from the blood pool. And then also some uptake in skeletal metastases and in the liver, which was where the qualifying lesion was in this particular patient. But it just gives a few examples of the localization that we see with this radiolabeled antibody. And next slide. All right, so this is 13 patients were actually eligible and assessed with dosimetry. Of those 13, 12 went on to receive at least 1 cycle of therapy. And so here are the radiation dose estimates presented for each one of the organs that is pretty much standard output for Olinda. We're using dose limits here for the kidneys, the liver, and the lungs. Those are specified in the protocol. So patients will continue to receive in the multi-dose portion of the study, the patients will continue to receive additional cycles up until they reach one of those limits. And also it's worth mentioning that we're using an RBE value for Actinium-225 of 3.4 for all these calculations. Next slide. And so just to call out a few of the radiation dose estimates, in particular, especially the ones that we do have those pre-specified limits. It shows with a single cycle we're quite far away from those limits with a single administration. And as we move along in our dose escalation cohorts, we see the expected increase in the exposure for these subjects. So also presented here a few organs and tissues that we don't have limits for, but they're worth noting as well. All right, next slide.
So in summary, just a quick perspective into our progress in the clinic thus far. Again, single ascending dose portion of our phase 1 study. And across the 3 cohorts of patients recruited thus far, all patients that were imaged qualified to go on to receive treatment with FPI 1434. An important part is the prospective and personalized treatment planning for targeted alpha therapy. And it's an important safety checkpoint, as Tom elaborated on, with respect to estimate risk for critical organs. The dosimetric results were well within the pre-specified single dose regimen up to the dose level of 40 kilobecquerels per kilogram. And in terms of the safety profile, as I mentioned earlier, both the therapeutic FPI 1434 and the imaging analog 1547 were really well tolerated without any dose-limiting toxicities or serious adverse events. So we're excited to continue enrollment. As I mentioned, we are currently enrolling patients into the multi-dosing portion of the phase 1 dose escalation study. And in parallel, we are conducting a cold subcohort analysis in patients, understanding, once again, the impact of pre-administration of naked antibody on targeted tumor lesion uptake of the imaging analog. So thank you for allowing us to participate today.
Thank you. That was a really encouraging presentation. Excited to hear more as it continues. So at this point, we'll move to our next presentation. So this comes from Neil Bander at Weill Cornell Medicine on dual targeting PSMA.
Greetings. I hope you can see my presentation here.
Yes.
Okay, great. All right, so we're all set. So continuing on the clinical theme, I'm going to talk about our work in prostate cancer targeting PSMA, prostate-specific membrane antigen, which I hope is familiar to most, if not all of you. And I'm also going to be talking about a somewhat unique approach that we're currently exploring in the clinic using dual targeting with both an alpha and a beta particle targeted by combining antibody with small molecule. Anyway, you'll see what I'm talking about shortly. As a Cornell faculty member, I need to provide you with my disclosures, and I do have conflicts related to the information I'm going to present today. And that is on listed on the current slide, including the fact that I'm an inventor on patents related to this presentation. And I do have, I am a paid advisor and a shareholder of a company that is developing this technology, which has been licensed from Cornell. So to start, my group has been interested for a couple of decades in the potential ability to target alpha particles to tumors in patients. This is a publication we had in Science, along with our collaborators, Mike McDivitt and David Scheinberg at Sloan Kettering. And in this publication, we showed in an animal model that we could quite effectively treat mice that were bearing human prostate cancer xenografts. But 20 years ago, this was not a clinical, it wasn't ready for prime time in the clinic. Some or many of you perhaps are aware that a few years ago from the University of Heidelberg, a couple of very interesting patients were published in a case report, which really triggered a lot of interest in the prostate cancer field and certainly reminded me of our previous experience with Actinium-225. And this is one of those two patients. I'll show you the other in the next slide. But what you can see here is a patient who began with a substantial burden of prostate cancer. His PSA was almost 300. He was treated with radioligand therapy initially with lutetium-177, and then ultimately got 3 doses of Actinium-225. And this patient had a complete biochemical as well as radiographic response. And it's worth pointing out in the upper right-hand corner, you can see that this is a patient who had exhausted essentially every form of known therapy for prostate cancer prior to his Actinium-225 treatment. This is the second patient in that report. This particular patient had a PSA of almost 3,000. Again, you can see virtually every bone in his body is involved with prostate cancer. And again, a complete biochemical and radiographic response to this treatment. But it's also worth pointing out, if you look at the red arrows on this patient, and if I toggle back to the last patient, what you see is, in addition to eradicating the tumor, because these radioligands also target the salivary glands and the lacrimal glands, you also eradicate those structures. And it turns out that that's a pretty intolerable side effect for these patients. In any case, when I saw those results, it occurred to me that in our biodistribution studies with antibodies that target PSMA, we do not get targeting of the salivary glands, and nor for that matter do we get excretion through the kidneys, which is another potential site of toxicity when you are targeting with small molecule radioligands. So it occurred to us that maybe we should harken back to the earlier publication and go back to the antibody and start clinically applying Actinium-225 in a clinical trial. And so this is the results of our first clinical trial. These results were presented by my clinical colleague, Scott DeGowa, at the recent ASCO meeting, I guess a couple of months ago. This was a single ascending dose phase 1 trial. There's a lot of data here, which I'm not going to walk you through other than to point out a few things. Number one, these patients are, as is typical in a phase 1 trial, were heavily pretreated. It's worth pointing out that almost half of these patients had already failed PSMA radioligand lutetium-177 treatment, and about 28% had also failed radium-223 treatment. So these were patients who had a significant radiation pre-exposure. There were 7 dose cohorts in this trial, which is shown in the upper right-hand corner. You can see the dose levels that were used. And it turns out that the treatment was very well tolerated. In fact, we did not reach an MTD. Only had 1 DLT, which occurred at cohort 6. And there were no DLTs at cohort 7. So we selected 93.3 kilobecquerels per kilo as the recommended phase 2 dose. This is, for the clinicians in the audience, you may recognize the PSA waterfall plot. What you can see here is the decrease in PSA experienced by the 32 patients in this trial. Almost 70% of the patients in this trial had some degree of PSA decline, with 44% having what we would consider a major PSA decline. It's also worth pointing out, I've already pointed out some of these factors to you. None of these patients were pre-selected for PSMA expression. That has led us to the current trial we have going on, which is a multiple ascending dose trial. We're exploring 2 different dose schedules, and this trial is in progress now. But for the second part of my talk, I want to describe to you what I think is a relatively unique approach that we're taking that I alluded to at the beginning of the talk, and that is this concept of dual targeting. And that's based on the fact that, really 2 facts. The first of which is that we knew the crystal structure of PSMA, which is shown in the lower left. It's normally a dimeric structure. We had previously epitope mapped the antibody to the apical region, which is shown in green. And we knew that the small molecule ligands that are used to target PSMA target the enzymatic pocket, which is spatially distant from the J591 antibody epitope. And we predicted that these 2 agents would not sterically interfere with one another's binding. And in fact, what you see on the right is that is in fact the case, that the 2 different colors represent 2 different cell lines. And you can see that when you add both reagents simultaneously, you get additive binding to the PSMA molecule. The other important feature that was necessary to pursue this line of clinical investigation is visually demonstrated on this side. And what you see in this slide is, on the left, you see the normal tissue biodistribution of the small molecule PSMA ligands. And on the right, you see the normal tissue biodistribution of the J591 antibody. These are both PET images. And what you can see is with the small molecule ligands, as I mentioned before, you see a lacrimal and submandibular gland uptake. You also see excretion through the kidneys. Conversely, with the antibody, you don't see either of those sites getting any detectable uptake by virtue of the antibody. So the key point here is that the normal tissue biodistributions of these 2 targeting agents is, for all intents and purposes, mutually exclusive. And in fact, that's borne out also in the side effect profile of these agents. They are, again, mutually exclusive. So this led us to what I've termed here our differentiated approach, whereas most, if not all others in this radionuclide targeting field are using either antibodies or or small molecule ligands. We thought that because of the 2 features that I just described to you, that we might have potential benefit of dual targeting by virtue of combining both the antibody and a small molecule ligand. And we anticipated that both of those agents would converge on the cancer, but not cause overlapping toxicity because of their mutually distinct biodistributions. And we'd have an additional benefit that each agent could carry a different payload. We've done a number of studies with this. Time is going to limit, obviously, what I can describe to you, but as one example, this is an animal model study where we're tracking both the antibody and the ligand and the combination thereof by virtue of SPECT-CT imaging in these animals that are growing prostate cancer xenografts. What you see in the orange, if you can see my pointer, is tracking the small molecule ligand over time, and it's just as you would expect, rapid and high uptake, which falls off over time. We follow these animals for 72 hours. You see the opposite with the antibody, again, just as you would predict, a slower but progressive uptake over time, as in part a function of the much longer half-life of the antibody relative to the small molecule ligand. The dotted line you see in yellow is what you would anticipate if we got additive binding to the tumor. But what we in fact measured by imaging was significantly greater than the sum of the parts, suggesting that the combination of the two actually was giving us synergistic binding to the tumor. Just to confirm that what we were seeing by virtue of imaging was in fact correct, we've done a number of studies where we actually autopsy the animals at 3 or 4 days after treatment, and we measure the number of counts per milligram of tumor. You can see that here in this first xenograft model. We again actually find it at 3 days post-injection, we have more counts per milligram of tumor delivered by the antibody than by the small molecule ligand. What we'd anticipate if we got additive targeting would be the sum of the parts, but what we actually measure, again by autopsy, is that we're seeing synergistic uptake by the tumor. And we've done this now in 3 different tumor models, and we see this in every single tumor model, that is synergistic uptake by virtue of combining antibody and small molecule ligand. We see this borne out also in efficacy studies. Here measuring the size of the xenografts, you can see in the green 617 lutetium is outperformed by the antibody actinium, and the combination of two actually gives us the best result. And I can tell you we autopsy the animals at the end of these experiments, and in the combined case, in a significant proportion of the animals, all we find left at the end of the experiment is the matrigel that was used to implant the tumors. So my assessment is that we're seeing 3 layers of benefit by virtue of the dual targeting. We see at a minimum, we get the additive dose to the tumor. We also see that the antibody improves the uptake and retention of the radioligand, and we've shown this in a number of ways, and I haven't been able to have time to show you that data, but that is in fact what we have determined. And the last benefit that I'll cover in the next couple of slides is the fact that adding the alpha to the beta particle gives us the benefit of the additional potency and precision of the alpha, but it also solves the limitation of the radioligand beta particle treatments with respect to treating small volume lesions. And that is in part demonstrated here. If you look at the absorbed energy as a result of delivery of lutetium-177, the smaller the volume of the tumor, the more radiation is delivered outside the volume of the tumor, and therefore the less effective lutetium is for treating these small volume lesions. And this, again, is borne out clinically. These are 4 different studies of patients who were treated with small molecule ligand lutetium, and the clear and consistent pattern of failure is in the development of what is referred to as new lesions, which are in fact subimageable lesions that were present at the time of initiation of therapy that are inadequately treated with the beta particle and then go on to become imageable over time. And this is again reflects the benefit of combining 2 different radioisotopes with different physical features. This is taken from what I consider a important publication by Joe O'Donoghue, showing that when you combine 2 different radiopharmaceuticals with different physical features, you can actually increase the curability range of the tumor size. And so we have initiated a study which started, we've dosed the first patients earlier this month, and it's a combination study of the antibody actinium plus lutetium labeled PSMA-INT in patients with metastatic castration-resistant prostate cancer. And I think that's where I'll leave my presentation. Thank you very much for your attention.
Thank you. I appreciate another really interesting presentation. All right, well, I will move on to our final presentation of the day. So George Sgouros from Johns Hopkins is going to speak to us now about dosimetry and treatment planning for Actinium-225 and other alpha emitters.
So I'm going to talk about dosimetry and the potential for treatment planning for actinium and other alpha emitters. So I do have some disclosures. The main thing I want to sort of promote is the idea that with agents that we can image and that we have surrogate imaging agents for, we're going to be optimally effective by moving away from what I call radioactive chemotherapy and taking advantage of the imaging and dosimetry capabilities to deliver these agents in a way that could lead to better outcomes. So it's sort of a hypothesis that needs to be tested, and there's been some testing of that. But to make this happen, we need to develop the tools, and that's what my group is focused on. So starting with imaging, and we can image the surrogates, but also it's useful to image the agents themselves, especially if you're going for a more than one cycle delivery approach to understand what's happening pharmacokinetically. So there are two approaches, right? We can do advanced reconstruction, and I don't know if anybody can see my mouse or, okay. So advanced reconstruction techniques are shown here, work done by Eric Fry. Essentially folding in the physics of the image formation process to generate quantitative SPECT images that can be done in a reasonable amount of time with the generally low administered activities that these patients are treated with because of the very high efficacy of alpha particle emitters. And here's sort of an example of these are images based on pharmacokinetic input for radium-223. They're sort of simulated images that are realistic in that the PK of the agent was input into a representation of human anatomy, and then the human anatomy was reconstructed and then used. So instead of using phantoms to understand the potential errors associated with quantitative imaging, this is sort of a more in silico kind of evaluation using simulated patient data. So we see reasonable uncertainties at different times with rapid clearance and reduced activity levels. And these are all for larger organs. When you get to smaller volumes, these uncertainties go pretty high up. Or you can build a new instrument. So this is work that Lin-John Meng at the University of Illinois, Urbana-Champaign is looking at to develop a CZT-based camera. I know there are some already available. This is a pretty sophisticated technique. What this is showing is the tremendously high energy resolution that one can achieve with these kind of devices. And the design that Lin-John has also incorporates clever ways of tremendously high sensitivity to account for the low administered activity. And this is sort of on the way, coming up in probably 4 or 5 years, a human system. The grant's already been funded to do that. But you can see some of the peaks here. So actinium can be, the peaks can be well separated. Here's francium, and here's some of the actinium peaks. And then the other, so we need imaging, and then we need a way to account for the fact that alphas have a very short range. So within the MERD committee formalism, right, we need to come up with S values that account for that very short range emission. So this is work with Wes Boltz and Rob Hobbs. Wes Boltz at University of Florida, Rob here at Hopkins, where we can think of macroscale based S values and then progressively go to lower, higher and higher resolution. So for example, in the liver, you can start talking about S values for a population of cells, potentially parenchymal cells or different structures within the liver. Kidney, same kind of thing. You can think of proximal tubule based S values, put them together to form an overall structure and nephron structure in the kidneys. So all of that then leads to these microscale S values, where one has an S value for a specific structure, and then within a tissue, this is for murine and for human, and then within a tissue, you put all this together, and you, if your agent localizes to the glomerulus, for example, then you have an S value for the dose from glomerulus to that tissue, and then you can see what that translates into if you have the whole kidney, irradiating the whole kidney, and so on. So this is in mice, and then the same thing for humans. And the reason for this is that this is half the aspect of doing a dose calculation. You also need to figure out how much of your agent is localizing into these different structures. And you can do that by pairing preclinical studies with human studies, and that's shown here. So in a human, with gamma camera imaging, you can measure the kinetics of the whole organ. You can do that in a human. You can also do it in a mouse. In a mouse, you can also look at the sub elements, and then by sort of just tallying up the different components, they should add up to the total organ. You can come up with apportionment factors that allow you to take a patient image for a whole organ, and then apportion that whole organ activity based on information you've obtained preclinically, which is kind of what is shown here. Alpha camera example, where we have, and this is work by Rob Hobbs and Anders here at Hopkins, actinium labeled antibody at different times, showing the different localizations over time. And then this later image here is most likely bismuth-213. And the other aspect with imaging is to fold in the daughters, and that can be done both with the instrumentation development and with the advanced reconstruction techniques, dual photon energy imaging. We heard a little bit about the RBE. Most RBE values are measured in vitro. Some are also in preclinical models. So we've also undertaken to do a rigorous evaluation of RBE values. So here's one of the issues that you have. You have a surviving fraction curve for cells on a petri dish. You incubate with different activity concentrations, and then you need to convert that geometry and activity concentration into an absorbed dose. That's easy for an external beam type relationship. You just irradiate the cells with an external source of radiation. If you're doing it with a radio-labeled antibody, in this case, this is a lead-212 labeled antibody, decays to bismuth-212, which is the alpha emitter. It's a bit more tricky, and I'll describe that in a little bit more detail later on. In vivo, this is sort of what we've been doing, looking at concentration in the different, and this is work from Joanna, Remco, and Rob, looking at the dependence on the percent injected activity per gram in the femur, the marrow, and in the blood for different levels of activity. And you need this to deliver different absorbed doses to get a dose-response relationship in vivo to come up with, and then related to what you get if you do external beam irradiation, to come up with an RBE for a specific endpoint. So take that data, put it together, combine it with, in this case, we're looking at marrow RBE, combine it with cellularity measurements, obtained both by increasing the activity concentration and also by external beam, just showing how we quantify this. And here's the table in terms of activity and cellularity in the femur region. And then you end up with something like this. So this is sort of work in progress, still being analyzed. Another thing I want to say about the in vitro assessments is, so external beam radiation is easy, right? It's uniform, whereas radiopharmaceutical therapy, one has to take into account localization and the geometry and so on. So when we do these calculations for in vitro work, we assume something like this. The reality, and these are slides from Renko Bastianet, who's working on this, a faculty member at Hopkins. This is the reality of what cells really look like in the dish, not exactly this nice grid pattern. And they don't just sit there, they actually move around a bit, they take up the antibody. This is a green fluorescent labeled antibody. And if we take all that and do sort of a Monte Carlo calculation for cells on a dish, we end up with something that looks like this. We take all of that information and we can come up with a sort of cell-specific or tissue culture-specific calculation of absorbed dose. And if we compare it to sort of the simpler kind of cells of the same size, equally spaced on a dish, we find that for membrane localization, the red is the MERD committee, the membrane, or the red is the simple sort of geometry-based calculation. The black is kind of the cell dish-specific calculation. And this is a histogram of the different S values. We find that for membrane localization, we're not that far off. And for cytosolic localization of the antibody, we're not that far off relative to the simpler model. When we look at the cross dose from one cell to another, there's a big difference. And really that's because cells aren't spaced apart in the way that the simple models assume. They're actually clustered together, and this really makes the big difference, which is great because it means that you don't have to do the full detail that Remco Bastianet in the lab is doing. If you can take some images of your cells and understand their spatial distribution, then you can potentially correct for that. So putting all this together, we're working on our own sort of clinical trial. In our case, it's a trial in dogs, in canine patients with osteosarcoma. We've been lucky. We've been able to get from Sloan Kettering, Nakhon Cheng, the humanized 3F8 antibody. It's an anti-GD2 antibody. And it turns out GD2 is also expressed on osteosarcoma cells. And we've shown by imaging, here's an FDG PET of Luke, our first dog, and maximum intensity projection SPECT image of indium labeled HU3F8, showing the lesions. And then on a coronal SPECT, again, you can see the lesions and sagittal and axial and so on. This is Jack, our second dog, showing big lesions. And this is the immunohistochemistry postmortem showing excellent staining in a lung met, and then also in the primary, the excised primary. So the plan is to basically run a trial that's driven by dosimetry in these dogs. We do FDG PET. We image with indium labeled 3F8 to get the PK. We do it 3 times. Using this, we specify the sort of optimal dose, maximum tolerated dose of the actinium labeled 3F8, toxicity monitoring, FDG PET again, SPECT CT again, and then standard of care. And the idea is to see to what extent a dosimetry-driven protocol will impact treatment of these dogs. It's not a multi-arm trial, but there's lots of data. And I know this is a big criticism for human study. There's lots of data on standard of care outcomes in dogs with osteosarcoma. They don't live long. And this is a collaboration with Yingli Fu, Joanna Liachu Yang Du, Jeffrey Bryan at Missouri, and Derek Reichman here at Hopkins. Some radiochemistry. So it turns out 3F8 is a little bit more involved in the labeling than the anti-HER2 antibody we've been using. And then this is acknowledging people in the lab, and then the collaborators, and also just highlighting, sort of advertising that we've started a Master's in Medical Physics program where part of the curriculum will focus on radiopharmaceutical therapy. Just a sense that we need more medical physics support. I don't know if everybody would agree with that, but I think it's important to really make it a multidisciplinary thing. And then this is the organizations that pay the bills. Thanks.
Well, thank you. I'd really like to thank all of our panelists for a set of fantastic presentations across a really broad variety of areas. So there's a number of questions in the Q&A. So we'll move to our moderated Q&A section. I guess I'll go through the ones that we've answered just to make sure that everybody gets to hear the responses. And then we can go to new questions. So the first question from Narissa Viola was there heating involved when labeling DOTA trastuzumab with Cerium-134? If so, what temperature and how was the immunoreactivity? Rebecca, are you? Do you want to go ahead and answer that?
I thought I answered.
Well, yeah, I don't know if everyone sees the Q&A, so.
Okay, yes, it was heated just so that we would be consistent with an actinium preparation protocol, and immunoreactivity was fine, so yeah.
Okay. The next question was from Henry Van Brocklin for Kate. How are the GP41 monoclonal antibodies crossing the blood-brain barrier?
Yes, so the human antibodies, it might be not a very well-known fact, but they have a isoelectric point, which is much higher than, for example, in urine antibodies, so which makes them cationic. And it's known that cationic compounds have a propensity to cross the blood-brain barrier. So our antibody, which we use for that specific HIV project, has one of the highest PI or isoelectric points of around 9. So basically it can gradually seep through the intact blood-brain barrier. And the reason for us trying to label it with Actinium-225, we also did the same for Lutetium-177 to have those long-lived isotopes, which would allow the antibody the time to go through the blood-brain barrier gradually, and the isotope will still be alive, will be around, so it could target the infected cells in the CNS. So that's basically it.
Thank you, Kate. The next question comes from Kandapa Baba. May I know what is the reason to incorporate PEG linker in between the DOTA and antibody in terms of clearance or therapeutic effect improvement, etc.?
Yeah, hi, this is Tom Armour. The PEG linker, from my understanding, really helps to modulate the clearance. So I think that's really part of the platform that Fusion Pharma is using to kind of control the kinetics of the molecule.
Great, thank you. And I saw this questioner also had a hand up. Did you have a follow-on, or can I unmute you? Okay, I guess we'll move on. Next question also for you, Thomas. Fascinating study with the Actinium-225 FPI 1434. Is there any estimate of tumor dose?
Yeah, so we get that question a bunch. You know, that's something that we're certainly looking at and expect to share with the community at some later point in time, but we haven't yet presented.
All right, thank you. Shesa, I think this question is for you. Can I ask if DOE is taking new customers for the thorium cow produced Actinium-225?
Yeah, unfortunately at this time, that product is fully subscribed and we are not able to accept new customers. If that changes, DOE will make an announcement. But for now, that's our status.
Great, thank you. Okay, so moving to the open questions for the FPI 1434 study, are you using thorium-229 derived actinium-225, or have you included the thorium-232 accelerator derived actinium-225?
Right
Yes.
now, oh, go ahead, Tom.
Yeah, go ahead, Jim.
I was just going to say, right now, our supply agreement with the DOE is for the generator-produced actinium, but we are, evaluating for, potential further use, the accelerator actinium as well.
Great, thank you. And then another question for Fusion. What drove the use of an RBE of 3.4 rather than RBE of 5?
Yeah, so we're actually using the 3.4 based on a reference. I think it's by Eldfist et al. And that really is our kind of reference point for the alpha. But I think it's really an unsettled question. We have some other data in-house that kind of lends us to believe it's within that range. But that's just the point that we picked and had kind of negotiated that with the FDA as well.
Great, thank you. I think that gets the next question as well. So we'll move on from Amos Het. For actinium, is there a reason you didn't set a limit for marrow dose in the patients? Typically, this has been 2 gray.
Yeah, so the marrow dose, there's multiple methods to do it. There's certainly a lot of uncertainty around that. So the patients are very, very closely monitored through the dose escalation for hematologic effects. So I think that's one area. Because of all of that, we were hoping to collect the dosimetry around the marrow, and then look at whatever toxicity we might run into, and then see what might be a more appropriate limit to set, possibly at a later point in time. So we weren't confident that 2 gray really was the limit in this particular case for actinium.
I would just add that, we have had discussions with the FDA about not setting that limit, and we have gained their endorsement to do so. As long as Tom pointed out, we're following the patients closely with weekly CBCs. We're looking for long-term toxicity. So there is a measure of comfort there.
Great, thank you. Another question. It seems no actinium-225 based radiopharmaceutical comes into phase 2 study. What is the reason behind this besides funding? Not sure who wants to tackle that one. Seems like phase 2 studies are on the near horizon.
Yeah, I would just say sit tight.
And I'm pretty sure for leukemia, anti-CD33 went beyond phase 2. Joe Jurczyk presented last year. I think he presented some phase 3 results. So there's nothing inherently wrong with actinium-225 that prevents it from going beyond. I think it's just a new agent, and it's a matter of time, as Neil was suggesting.
Well, we certainly look forward to more continuing studies. Question from Michael Nichols. In general, performing quality control, especially HPLC, is difficult for actinium-225 drug products. Can Thomas comment on if this is done for their product, and if so, what is your approach?
Yeah, so I can't really say. I'm not really on the manufacturing side, so I don't know if I can really speak directly to that. But I know that it does undergo quality evaluation before release and delivery to the site. But as far as all the details of that, I don't know that I can answer that. Apologies.
Yeah, perhaps that's a question. If there's interest, you can reach out to either Tom or myself, and we can follow up with the appropriate CMC group here at Fusion.
Is anyone else on the panel used HPLC to maybe want to speak up? Or Kate, yeah, thank you.
I can say that in our experience, HPLC is not very useful for actinium labeled products because actinium is constantly decaying, and you instantly in your radioactive trace, you will see francium bismuth. So it creates the impression that your product is not pure, even though you have purified it. So I think HPLC should be performed when the labeling procedure is still being worked out. But as a final QC, we don't find it very useful.
Okay, thank you. I'm going to skip over to the chat. There's a question in the chat from Naikong Chung. Regarding high PI and crossing of the blood-brain barrier, HU3F8 has a PI of 9 to 10, and we could ask Dr. Sgouros if he sees any penetration of his Actinium-225 conjugate into the canine brain.
So it's the Indium-111 that we've imaged. We haven't seen it in our dog studies.
Okay, thank you. Back to the Q&A for Fusion. If you use both generator and accelerator produced material, do you plan on mixing the two in a single study? Do you know if the FDA will accept both sources in a single study?
I think the short answer to that is very good questions, which no one probably knows the answer to today. But definitely part of our thought process should we go down the road of using the accelerator produced actinium.
Agreed. Does anyone else on the panel want to weigh in general? Okay, we'll move on then. What are the panel's thoughts on the redistribution of daughter radionuclides and the potential toxicities that might result?
So I'll take a stab at it. I think it depends very much on the agent, and the reason it depends on the agent, because it depends where the decay happens. If it happens in the circulation, then you do have free bismuth-213 in circulation and rapidly going to the kidneys. If it happens within a tumor cell, the odds are that the bismuth is going to stick to something within that tumor environment. So it's not, and the implication of what I just said is that it depends on the agent. Agents that have a long circulating half-life are going to have more daughters in other organs compared to agents that clear quickly, agents that localize rapidly to tumors. So in a sense, you can establish the parameters of how to think about the problem, and then maybe do some pharmacokinetic modeling to add indium or zirconium-based dose calculations to account for the daughters, bismuth-213 in particular.
Would any other panelists like to weigh in? Okay, well, that's all of the questions. Guess I'll have a last call if there are any other questions that people would like to put into the chat.
While we're on the call, can I ask Neil a question?
Sure.
Neil, your study design is really intriguing to use both the alpha and the lutetium. I'm wondering if you're at liberty to tell us kind of the sequence of administration, and do you deliver them concomitantly, separated, and what a cycle length is, or what you consider a cycle?
Yes, so we actually administer them simultaneously. And I'm trying to remember the cycle. So I think the cycles we're using are every 6 weeks. Is that answer your question?
Yeah.
Yeah.
So kind of,
But I think it's worth pointing out, I should add, we don't really know what the optimal cycle is. And in one of the trials that I showed on the slide, was in our current protocol of multi-dose single agent antibody Actinium-225, we're looking at 2 different dosing schedules, one of which I would call the more classic or standard cycle of every 6 weeks. But we're also looking at a more dose intense regimen where patients get 1 cycle of 2 doses given 2 weeks apart. We've done that with 617 lutetium in a study of 44 patients. And we've also done that with the antibody lutetium. And there's reason to think that the shorter and more dose intense cycle is as good, if not better, than multiple doses given over every 6 to 10 weeks. But we're looking at that in that trial. We have both arms going simultaneously in parallel, and once we finish that trial, and both of them have expansion arms, so we'll be able to do some degree of comparison of the 2 different dosing schedules.
very good. Interested to see those results when they're available.
So am I.
Okay, well, if there's no further questions, I would really like to thank our panel again for their great presentations, and I would like to thank everyone for attending. I think it's been a great discussion and a great day. So there will be additional user meetings for other isotopes. Those are advertised on the NIDC website. And with that, I guess we'll sign off. Thank you all again.